Yeah, hello everyone. Very happy to be here. I'm Sarah. I'm a postdoc in the Neural Control of Movement Lab at ETH Zurich, and I would like to tell you something about the pupil-based biofeedback approach we developed over the past years, where we try to teach participants how to self-regulate their arousal levels. Why are we interested in that? Arousal is actually quite crucial for our well-being, how we behave, how we feel, for our cognition. So for example, what has been shown is that if arousal levels are aberrant, they're tightly linked to stress-related disorders, but also sleep disturbances. And one of the crucial regulators of the brain's arousal level is the locus coeruleus. So it's a small structure deep in the brainstem, in the pons actually, and it's the major source of noradrenaline in the central nervous system, in the brain. So when the locus coeruleus is active, so for example, when we encounter a stressful situation, it would release noradrenaline, and we have, for example, increased cortical activity and also effects, for example, on heart rate and so on. And the problem is, its position, it's very deep in the brainstem, it's not so easy to monitor it or to even modulate the system in a non-invasive way in humans. What has been shown in previous studies, also in studies in our lab by my colleague Valerio Cerbi and his team, is that pupil size is actually a nice readout of the brain's arousal levels, mainly driven by the locus coeruleus, not only obviously, but the LC is a crucial key hub here. So before LC activation, they see, as you can see here in a mouse, so the pupil is very small. If they activate the locus coeruleus chemogenetically, you see a drastic pupil size increase. So what we basically did is to translate this link into a new tool where we basically measure pupil size changes. We feed them back in real time to participants, and like this they can basically develop mental strategies that allow them to basically target the brain's arousal levels or the LCNA system, which is then reflected again in pupil size changes. First of all, we wanted to know whether this is even possible. There have been some studies showing that people can actually volitionally dilate their pupil in one session, for example, also over multiple sessions, but especially the decrease was never so consistently. So we developed a pupil-based biofeedback training over 3 days, and then we looked whether participants can learn to increase, so to upregulate and to decrease their own pupil size. And this is indeed the case. So here's the biofeedback group. They nicely show an upregulation, so an increase as compared to a baseline measurement, and also decrease over the time course of 15 seconds. In a control group, they had the same amount of training, same information, but no valid feedback. We see that it's slightly and significantly reduced. So it seems that we can actually indeed learn to volitionally change our own pupil size via mental strategies. The next question we had is, of course, what is happening while we actually do that? So we went into the MRI and we did brainstem fMRI to look at bold activity changes during up and down regulation of pupil size, and we're specifically looking at the locus coeruleus. And for everyone who does brainstem fMRI, it's not so trivial because it's very sensitive to physiological noise, so we had to correct quite a bit for it. For example, we have to get rid of respiratory and cardiac signals, which is, of course, also heavily linked to our signal. So I guess we took out also some signal of interest here, but we had to do it to get rid of the noise, basically. And what we nicely see is if participants increase and decrease their pupil size, there is indeed a higher activity in the locus coeruleus during up as compared to downregulation, but we also see it, for example, in the dopaminergic system, so substantia nigra and ventral tegmental area, and similar but slightly reduced effects in the cholinergic system, so in the nucleus basalis of Meissner. Next, we wanted to go a step further and look into EEG to go more to the cortical markers of arousal, and the 1/f slope was really nicely introduced. I guess conceptually I don't have to say so much about it. I mean, we also based it on sleep studies, for example, where you could see that you can nicely distinguish between different sleep stages with the slope, so it gets steeper in deeper sleep stages, for example, as compared to wake. And we estimated the slope for a frequency range of 30 to 40 5 hertz. We also did it for 1 to 40 hertz. It was pretty similar results. And what we see is that the slope gets indeed steeper during downregulation, so when pupil size is smaller. And crucially, this was not the case during our baseline measurements before the modulation, but it was basically only the case when participants would downregulate. And these are, again, the 15 seconds of down and up regulation. When we look into cardiac signals, just very basic, because we just averaged heart rate for up and down regulation and also looked into RMSSD, which is the only measure we for now took, because we have these very short 15-second time windows, which is crucial to, or which is difficult to take other measures to look into it. And here we also saw that heart rate is higher for up versus down regulation, and RMSSD is lower for upregulation as compared to downregulation. Also, if we removed this participant here, it still stayed the same. Next, we went to, again, a different method, basically, which is mainly led by Veronika Potok-Szybinska, who's also here. So basically, what she did was to combine the pupil-based biofeedback approach with TMS, and as we've already learned earlier on in the keynote lecture, we have motor-evoked potentials. So basically, when we stimulate over the motor cortical areas, we elicit MEPs, and if the LC is active, and when it releases noradrenaline, for example, then we would expect that this facilitates cortical excitability. So we wanted to test whether we actually see this when participants self-regulate their own pupil size, which we have previously shown to tap into this LC activity. And this is indeed the case. So during upregulation of pupil size, participants have higher MEP amplitudes as compared to downregulation when we normalize it to a resting control condition. And this was not only the case for, so give a little spoiler here, so for upregulation, participants can, for example, use emotional-related strategies so that they would imagine emotionally relevant situation, but they can also go more into physiologically challenging imagery, for example, that would also increase pupil size. And we saw it for both, for different strategies, basically an increase in excitability. Then, leaving with the physiological and neurophysiological side, we, in our last experiment, we combined it with emotional or neutral sounds, so especially negative sounds, to see whether when we self-regulate pupil size, we would actually see a change in the self-ratings towards the sound and also in physiological ratings. For the interest of time, I would only spend the last couple of minutes on the self-ratings of effect, so the intensity of the experience effect the participants report. And what we basically did was we trained our participants to do pupil self-regulation, then they came back, and in a separate session they did simultaneous self-regulation while we presented sounds. And then we were basically looking into these signals, and we're looking whether our self-regulation and also our training is doing something. And what we found here, and bear with me, unfortunately the transition to a Windows computer has removed the confidence interval, but I think you can still nicely see it. What happens in general is that if we look into especially the negative sounds, our training self-regulation success, and especially downregulation, upregulation did not contribute to it, was linked to the effect rating. So basically participants who could profit more from the training, so who could better downregulate at the end of the training as compared to the beginning of the training, which is reflected here in negative values. They have lower effect ratings during downregulation, also during the control condition, but not during upregulation. So what we don't know by now is whether this is more like a inherent trait, that participants who are less affected by these negative sounds are also simply better in self-regulation, which is also reflected in pupil self-regulation, or whether it's really a training effect. So we have to investigate this further. So to quickly wrap up, we see that we can actually indeed achieve pupil self-regulation. We see that it's linked to LC activity changes, also to changes in other arousal regulating centers in the brainstem, so dopaminergic, cholinergic. We see changes in cortical arousal markers, so 1/F slope, and we also see changes in cortical excitability as well as cardiac responses. Regarding the emotional stimuli, I think there is still some discussion going on what we really see here. And with this, I would like to go to some future plans. What we were also thinking about in the summer school here is really to look also into respiratory signals, because we record it, and I think it would be really nice to look into it. And also inspired by Diego's workshop into the sympathetic and parasympathetic changes in our cardiac signals. With that, I would like to thank the NCM lab in Zurich and also the HMZ flagship project Stress, where we involved in with the pupil-based biofeedback. And I would like to thank you for your attention.
Thank you. Wonderful talk. Are you aware what kind of strategies the subjects use to regulate your pupil size?
Yes, we are. So we do a proper debriefing at the end that we really know what they are doing. And for the upregulation, I think I showed it in one of the plots, a lot of people, they really go into, I would say, emotional imagery, if I can describe it like that. So they would really vividly imagine emotional situations. For example, some rather go into physical challenges, for example, that they imagine they have a very steep hill in front of them and they need to kind of overcome this challenge, which is also tightly linked, for example, in animal studies with LC processes. For the downregulation, some people, they can easily do a blank mind after training, which is, I think, a very good skill to just not think of anything. Some do relaxation-based imagery that they think of, really things that relax them in a way, yeah.
Do you make suggestions, or do they simply come up with that?
Yes, in the data I showed here, it's really suggestions that we give them based on previous literature, but we also now ran a pilot where we didn't give any instructions at all. We basically said they should increase and decrease the signal they see, and it works equally well, so that's nice.
Maybe in a similar line of thought, I mean, could that be something about the effects that are just general for figuring out the hidden rule in this kind of biofeedback task? So if you, for example, would have participants train to increase alpha or downregulate alpha, because, I mean, just to check which of those effects are really kind of specifically targeting the areas you're most interested in, and what are global effects of kind of modulating arousal or kind of navigating in this unknown rule space.
Yeah, I think we see rather, I don't know whether I understood your question correctly, but I think we see in general global effects of arousal as well. Because I mean, also the pupil, of course, it's a very, I mean, it's very strongly related to the brain state you're in, right? And when we, for example, look at the slope, it's also very global change. We, for example, observe when we link the two measures, it's correlated, yeah. But yeah, other than that, I don't think I can give more. But maybe one thing, yeah, what we are now doing basically is also look a little bit more into a different strategy, whether it has more specific effects. But yeah, I don't know whether you were targeting this, yeah.